Module 2, Bacterial Diseases and Diagnostic Methods. Lecture 1, Symptoms of Plant Diseases Caused by Bacteria. Bacterial pathogens, though smaller and less diverse than fungi, cause significant plant diseases. Symptoms often include leaf spots and blights, water-soaked lesions that turn necrotic, wilts, vascular tissues clogged by bacterial slime, cankers, sunken, dead areas on stems or branches, galls, abnormal swellings, such as crown gall caused by Agrobacterium tumefaciens, soft rots, tissues broken down into watery masses by bacterial enzymes. These symptoms often resemble fungal diseases, so proper diagnosis is essential. Lecture 2, Isolation and Identification of Pathogens. Accurate identification of the causal agent is critical for disease management. The process begins with isolation. From an infected plant, samples of diseased tissue are taken and cultured on nutrient media. For fungi, common media include potato dextrose agar. For bacteria, nutrient agar or selective media are used. The pathogens grow into colonies that can be observed, characterized, and tested further. Identification may rely on colony morphology, microscopic features, biochemical tests, and modern molecular methods like polymerase chain reaction. Lecture 3, Obtaining samples from infected plants. Proper sampling is the foundation of diagnosis. Samples should be taken from the advancing margin of disease, where healthy and diseased tissues meet. This ensures the pathogen is actively present. Samples must be collected carefully to avoid contamination and transported in sterile conditions. If improperly collected, results may be misleading, leading to wrong conclusions about the cause. Lecture 4, Preparing media for culturing pathogens. Media preparation is a key laboratory skill. It involves mixing the appropriate nutrients, sterilizing the medium through autoclaving, and pouring it into sterile containers. For fungi, carbohydrate-rich media supports spore germination and mycelial growth. For bacteria, media often contain peptones and other nutrients. Some media are selective, allowing only certain organisms to grow, while others are differential, highlighting particular traits. Care must be taken at every step to maintain sterility, otherwise contaminants may outgrow the pathogen. Lecture 5, Procedure for Diagnosis of Fungal Diseases. The diagnosis of fungal diseases involves several steps. Field observation of symptoms and signs. Collection of samples for laboratory study. Isolation of the suspected pathogen on culture media. Microscopic examination of spores, hyphae, or reproductive structures. Pathogenicity test, where the isolated fungus is reintroduced to a healthy plant to confirm it causes the disease. This systematic approach ensures that the true causal agent is identified. Lecture 6, Procedure for Diagnosis of Bacterial Diseases. Similarly, diagnosing bacterial diseases involves observation of water-soaked lesions or ooze, isolation on selective media, biochemical testing, such as sugar fermentation or enzyme production, microscopy, often showing characteristic bacterial cells, pathogenicity testing on healthy plants to confirm. Advances in molecular biology now allow rapid DNA-based identification, improving speed and accuracy. Lecture 7, Importance of Accurate Diagnosis. Why is diagnosis so critical? Because control measures depend on knowing the causal agent. A fungicide will not control a bacterial disease, and antibiotics are rarely used in plant pathology. Misdiagnosis wastes resources and may worsen outbreaks. Accurate diagnosis allows targeted interventions such as resistant varieties, cultural controls, or appropriate chemical measures. Lecture 8, Integrating Knowledge for Disease Management. Ultimately, the purpose of studying pathogenic bacteriology and mycology is to apply this knowledge in disease management. Farmers, extension workers, and researchers collaborate to monitor fields, diagnose problems, and recommend solutions. Integrated disease management combines cultural practices, resistant varieties, biological control, and chemical measures when necessary. The aim is sustainable crop production and protection of food security. Module 2 Summary. In Module 2, we focused on bacterial diseases and diagnostic methods. We examined symptoms of bacterial infections, steps in isolating and identifying pathogens, sampling methods, media preparation, and procedures for diagnosing fungal and bacterial diseases. We emphasized the importance of accurate diagnosis and integrated disease management. Course conclusion. This course on pathogenic bacteriology and mycology introduced you to the principles of plant disease development, historical impacts, fungal and bacterial pathogens, and the essential laboratory and field techniques used in diagnosis. Module 1 established the fundamentals, focusing on the concept of plant disease, the disease triangle, fungal classification, and symptoms of fungal diseases. Module 2 expanded into bacterial pathogens and practical diagnostic methods, including isolation, culture, and identification. By the end of this course, you should appreciate how plant pathology underpins agricultural productivity and why continuous study is vital for protecting crops and ensuring global food security.